Welcome everyone to our monthly precision medicine seminar. It is my pleasure to introduce Dr. Ruth Loos, who is a Vice Executive Director of the Novo Nordisk Foundation Center for Basic Metabolic Research at the University of Copenhagen in Denmark. She's calling in late in the evening from Denmark. She also at the same time serves as the Director of the Genetics of Obesity and Related Metabolic Traits Program in the Charles Bronfman Institute of Personalized Medicine at Sinai. Ruth is an internationally recognized leader in studies of obesity, genetic studies of obesity and related metabolic traits. She has been really involved in the genetic discovery studies for these phenotypes right from the inception of GWAS methodology. In 2005, she joined the MRC epidemiology unit of the Institute of Metabolic Science in Cambridge in the UK, and she became a group leader of the genetic epidemiology of obesity program at Cambridge. She then led several of the landmark discovery GWAS studies for BMI and metabolic traits. She was working closely within the GIANT consortium. And in 2011, she joined the Sinai team where she continued her studies of obesity, lipids, glucose, insulin, type 2 diabetes, blood pressure, and related traits. More recently, with a focus on ancestrally diverse populations and perhaps on a rarer spectrum of genetic variation. She's also interested in gene-environment interaction and their role in shaping susceptibility to metabolic syndrome and obesity. So I'm really excited to have her speak at our seminar and to learn more about her latest work in the field. Welcome, Ruth.
Thank you so much for the introduction and for the invitation to speak. So my title is on the genetics of obesity, and want to tell you for this talk a little more about what we've learned in the context of biology and how that may impact also precision health in general, or at least in the context of obesity. Let's see. So probably don't have to tell you, but obesity is a serious public health concern. And this graph is often shown to make the argument that obesity is really the result of a changing environment. So this is the prevalence of obesity in 1975. 5% of the population was had obesity. And that's what you see here in green. And as the years went on, the map colored more and more red. And by 2016, 12% of the population had obesity. And the World Health Organization estimates that every year, 2.8 million people die because of the consequences of obesity and overweight. And of course, it's not a competition, but just put it in perspective. In the last 2 years, each year, 2.8 million of people have died of COVID. So just to put it in perspective, give you an idea of the magnitude of the problem. But obesity is more than just a changing environment. It is actually the people who are genetically susceptible to this changing environment who will gain the most of weight. So indeed, twin and heritability studies have shown that there's a good component of the variation between individuals in BMI that's due to genetic variations. Like 40 to 70% is the typical estimate that we get from twin and family studies. So that's a reasonable amount of individuals. And indeed, if we look at the results of the GWAS studies, this is what the current status is. So GWAS started in 2007 with the first discovery, and the scale is out of proportion these days, but it was one locus. And as sample sizes increased over the years, the number of loci, the number of discoveries increased as well. And I can already tell you that, okay, by now we have more than 1,000, almost 1,200 loci identified, but the GIANT consortium is currently working on a bigger meta-analysis. GIANT combined data already for 2.1 million and has just added another 3.5 million individuals for overall meta-analysis of 5.6 million people. So you can imagine that this number of 1,200 loci will soon, I think it will at least triple, if not more, When you get to these numbers, it becomes very hard to decide where a locus ends and where a new locus starts. It's almost like the whole genome is associated, which is of course tricky. What you see here, similar to many other complex diseases, is that the vast majority of the loci have first been identified in individuals of European ancestry, much fewer in Asian, and even fewer in individuals of Hispanic or African ancestry. What you also see here, and this is the same data, the vast majority of loci were first identified for body mass index, whereas far fewer of the loci have been identified for other adiposity traits. And this is of course because body mass index is easy to collect, weight divided by height squared, which means that almost every study with GWAS data has body mass index. Meaning that the sample sizes can be big, the power to identify loci is big as well. But at the same time, body mass index is a very crude measure. And I'll come back to that later to comment on that again. So the question is, what have we learned from all these genetic loci, like so far more than 1,200? So the first reasons for people to do GWAS was mainly to gain insights in the biology. So using genes to study the underlying mechanisms of, in this case, body weight regulation. But as more and more variants are being identified, we're also using these genetic variants in the context of prediction studies, causality, Mendelian randomization, and so on. So what about the biology and mechanisms? So already very early on in the GWAS era for obesity, we've seen that the brain always turns out to be a key organ in body weight. So if we do tissue enrichment analyses, we take the genes within the obesity associated loci and we compare them with the gene expression of genes outside the obesity loci, then it's always the brain that seems to be enriched in the obesity associated variants. And this is an example of the PICT analyses, tissue and cell type enrichment analyses, but whichever way you analyze it, here again, the CNS shows up in this analysis. Again, the brain here, CNS shows up here. And if you do pathway gene set analyses, it's again pathways that act in the brain, and probably through controlling controlling the hedonic aspects of food intake, hunger, reward, satiety, and so on. And that's very consistent with what we know from the monogenic forms of obesity, where obesity is really caused by hyperphagia and not so much like lower energy expenditure. But that's of course broad biology. We really want to dig deeper into the deeper layers of the biology. So to do that, we really have to translate variants into genes, into new biology. And as with any other complex disease, it's also been hard for body mass index and obesity. Just to give you an example, this is the FTO locus identified more than, how many years is it? 15 years ago, more than. Then you see here a very nice association signal, chromosome 16 in the intron of FTO. And early on, it seemed like that was in 2007. Early on, we really believed that FTO was the causal gene, and many good studies, or I should say papers published in good journals, came up with a mechanism how FTO could be the causal gene. And these mechanisms were all different. So here's just 4 of the prominent papers published back then, all actually pointing to very different potential biology. And soon thereafter, people started thinking like, maybe FTO is just an innocent bystander. Maybe we've been misled by that high association signal, and maybe there's just communication between this intron and genes nearby. And indeed, George Stratigopoulos from and Rudy Leibel's group early on already showed that this gene RP group 1L could be the causal gene, and RP group 1L encodes the basal body of the primary cilia, which by virtue of similarity with the Bardet-Biedl syndrome that has obesity as a feature, could indeed be a causal gene for obesity. but then others thought like, well, maybe it's not FTO, but maybe it's also not RP group 1L, but the action may be on the other side of the gene. And they thought it was IRX3 that could be the causal gene through projections, through examining the regulatory effects of this variants in this locus. And one paper thought that FTO, or the FTO locus influenced IRX3 through acting in the brain, influencing hunger and satiety as well. Whereas the other paper thought it had more, or the other study thought it had more to do with the browning of the white tissue, which would lead to like more energy expenditure, which would affect body weight. So even though they each pointed to the same gene, the mechanisms that they proposed were different. And then just very recently was actually some sort of a unifying paper in Science where they said, well, maybe it's IRX3, but maybe it's also IRX5, and maybe RP group 1L is involved, and also the mechanisms in both the adipose tissue and the brain could all be involved. So it seems like this paper suggested large-scale pleiotropy at this locus. But this is just to say that even those signals can be very clear, it's still very hard to truly identify what's the mechanism, what's the causal gene. So I would say, like, even for FTO, it's still not totally clear what's going on, and that's been 15 years. Then over the years, a few more studies have been published that have shown some credible gene prioritization and new biology. And here I'm showing you 5 studies, and these are not examples. These are actually the 5 studies that I could find that really had gone sort of into like a more deep molecular analysis. It's like in one, the first study focuses on TMEM18, the other one on CADM1, CADM2 here as well, ADCY3 and NEGR1. And these are pretty much the studies that I could find with some robust and validated findings. And remember, we have more than 1,100 loci identified, so there's a lot more of the loci that remain to be elucidated. So what we did in my group, or what we're actually still working on, is like, let's use some bioinformatic tools to prioritize genes within the BMI associated loci. And our starting point was one of the latest papers by the Giant Consortium that combined data of 700,000 individuals and that identified 536 of the BMI associated loci. And this is a Manhattan plot. You barely see that's a Manhattan plot, but you see the p-values go to, I don't know, 10 to the minus 2000 or something. And that's here for FTO, barely visible. The red line is the significance threshold of 10 to the minus 8. So you can clearly see that everything is out of scale here. Anyway, we tried for each of these loci to identify what might be the causal gene, and we used 8 different tools. And this is like a summary of the tools, 6 locus-based approaches, including MAGMA, FineMap, MSC, SMR plus HIDE, TWAS, and the nearest gene, and then 2 similarity-based approaches, DEPICT and POPS. And what we did is we identified for each locus which gene was being prioritized by each of the methods, and then simply added or counted how many times the same gene was being prioritized. It was a little more refined than just adding or counting how many times a gene was prioritized. We multiplied, or what we also did is we had a set of gold standard genes for obesity, like MC4R, POMC, BDNF alpha, leptin receptor. And if one of these 8 approaches was able to identify these genes, then they were weighed more than if an approach was not able to identify. So we built in a weighing factor that was like included in the counting. So by doing so, we identified several genes that had been identified multiple times. So maybe you've seen figures like this. It's called an upset figure. What you see here is the 8 methods that we use, magma, nearest gene, POPs, and so on. And here on the right-hand side, it basically tells you how often each of these methods have prioritized a gene once. Yeah, so magma is the most greedy approach. Magma identified 1,578 genes that had not been prioritized by any of the other methods. So it was the only method that identified these genes. Then POPs identified 290 genes that only were identified by POPs and none of the other methods. And that's of course not interesting. You want that genes are identified, prioritized by multiple methods. Yeah. For example, 383 genes had been prioritized by 2 approaches. Just for clarity, like 95 genes had been identified by magma and were the nearest gene. 82 genes were prioritized by magma and POPs and so on. So the more we move to the left, the more interesting it gets. Like 86 genes were prioritized by 4 approaches, 34 by 5, and 6 genes were prioritized by 6 out of the 8 approaches. And just to give you an idea, I won't go into all the details because it's basically a list of genes, but these are the 6 genes that we prioritized. And some of these are already good, plausible candidates. And just thinking of this ANKRD26, it had been shown several times, but several times by the same group, that inactivation in mice results in hyperphagia, extreme obesity, gigantism. And that's, of course, encouraging. That was already identified in mice, and now we prioritize that gene in humans as well. CAT8, Jackie Stevens at LSU in Pennington showed that CAT8 is involved in adipocyte differentiation. And this last gene here, it's like said that it also plays a role in adipocyte differentiation. Just to give you an idea, is like what the results are from this approach. But again, this is only the beginning of like further translation. And we're actually, if any of these genes are of interest to you and you want to follow up, please let me know, because we're looking for more functional data to finalize the paper here. So since this translation from locus to biology is so hard, mainly because many of the variants are not located in genes, but outside genes, we thought like maybe it is easier if we just go straight to the protein altering variants. Yeah, we focus on low frequency variants that result in amino acid change and loss of function and so on. So that was a study that we did a few years ago, also with the GIANT consortium. We didn't have enough money to sequence large cohorts, but we did have enough money to buy a chip that was based on sequencing data, so that captured rare variants in protein, rare protein altering variants. So this chip included 216,000 variants. And you see here the distribution of missense, splice, and stop gain variants. And we had data of almost 720,000 individuals. So it's an enormous cohort. At that time, that was the largest cohort that we ever had with GIANT. So we had expected that we would have good power to identify these rare variants. And in a way, we did have power to identify variants, but not that many, actually. So we had identified 14 variants in 13 genes. And that's what you see here, the Manhattan plots. Some credible candidates, such as MC4R, GIPR, and so on, but also some new ones that showed up. And again, some of these new ones show plausible insights in the biology, but at the same time, several of these variants were actually not rare enough to be interesting enough. And that, of course, has to do with the fact that we still used a chip that was based on 10,000 individuals and that we did not do sequencing in these 700,000 individuals. So we thought, like, actually what you really need is sequencing in a huge population. And that's indeed, last year what happened is that the Regeneron, they indeed sequenced the exomes of 740,000 individuals. So almost the same sample size as we had a few years ago. But in the end, they didn't identify that many new variants. And you might argue that maybe the number is not important as long as the genes that you identified are biologically meaningful. So what they did is they had data mainly from the UK Biobank, but also from the Geisinger and from a cohort in Mexico. And they did gene burden analyses for rare non-synonymous variants. And or at least 16 of these across the whole genome, 16 genes showed a significant association with body mass index. And these 16 genes. So what you see here is the allele frequency on the x-axis, and on the y-axis the effect size of the association. In blue are the sort of the more common variants, whereas in orange are the variants that they identified. GPR151 and GPR had been identified before, MC4R had been identified before, but then they have identified a few new ones. And in their paper, they decided to focus on this GPR75. And as you can see, it's associated with a lower body weight. And of course, if you have a GPCR that has a protective effect, that makes it probably interesting for a drug company. And indeed, when they knock down this GPR75 in mice, then it results in a lower weight gain. That's sort of one of their first functional studies that they show here. What's important is that it's very rare. Only 1 in or 4 individuals in 10,000 carries a non-synonymous variation in this GPR75. And if you carry such a variation, then your body mass index is about 2 BMI units lower than in someone who does not carry a variation. And that is actually a lot. 2 BMI units is a lot. When the paper came out, there was a lot of attention of the press, and they made it sound as if, like, if you carry a variation in GPR75, you will always be normal weight. But actually, the paper itself clearly showed that that's not the case. So this is actually an interesting figure where they first show here in the middle is the distribution in the majority of the population, 645,000 individuals, of their normal weights, overweight, obese, and extremely obese. And this is the distribution when you carry any of the variations in the GPR75. So 228 individuals out of the 650,000 individuals carry a variation in GPR75. And indeed, far more often they are normal weight, but there's still like more, the majority is actually still overweight or obese. Yeah. So that's actually an important finding, that the penetrance of these rare variants, they're actually very rare, is definitely not complete. So there must be other factors that make carriers of these GPR75 mutations still be overweight or obese. I thought it was also interesting that they showed this graph for MC4R. So MC4R is well known that mutations in MC4R, certain mutations in MC4R cause obesity. So they're associated with increased risk. And again, they indeed show that carriers out of the 324, the majority have obesity or extreme obesity, but there are still carriers of MC4R mutations that are normal weight. Yeah. So again, while in the past these have been called monogenic forms of obesity, it's hard to say that it's really a monogenic form of obesity. And in fact, we had a paper a few years ago where we showed that these individuals who carry MC4R mutations but who are normal weight, their polygenic background seem to be more protective than average. And that may be one of the reasons why the penetrance is incomplete, so that there's an interaction, or it's like a canceling out of the monogenic susceptibility through polygenic protection. So this is sort of the main insights that we got from the biology. So I should say, to sum up our insights in the biology, we know that the brain is involved, but beyond that, the studies have not really revealed big breakthroughs in terms of biology. And of course, these things take time, but I think we still need much more work and hopefully much more interest of molecular biologists to follow up on these loci. And as I mentioned, as more and more variants are being identified, these variants are being used in polygenic scores to do prediction and so on. So a few years ago, a paper by Amit Khera came out entitled Polygenic Prediction of Weight and Obesity Trajectories from Birth to Adulthood. So it is very interesting and it caught a lot of attention. The press was on it. They even a few months after their paper came out with a chip, $50. You could, for $50, you could your genotype be tested for your susceptibility to obesity. Then the question is like, how good was their score really? Because they used a score to predict obesity. Well, over the years, of course, their attempt to predict obesity was definitely not the first. Like over the years, we've been trying to predict obesity. Like just to give you a few numbers, in 2007, FTO, the first locus was identified, and because the association signal was so strong, we thought like, wow, now we can predict obesity. But the reality is that the FTO variance explains only 0.34% of the variation in body mass index, like not 34%, not 3.4, but 0.34. So if it explains only that, it's impossible that it's going to be a good predictor. And indeed, if we calculate the area under the ROC curve, then you see that it's 0.55 basically. So it's just a little better than flipping a coin. Yeah. So this curve goes normally between, I would say between 0.50 up to 1. And the closer you're to 1, the better your prediction is. Like ideally for clinical use, you want that the area under the curve is 0.8, even 0.9 be better. So with 0.55, we're really far off. But then over the years, we added more loci, but the predictive ability not really improve. 2009, 12 variants, 2010, 32 variants. We go from 0.55 with FTO to 0.58, not meaningful. 97 in 2015, we explain out 2.7%, still not a lot. And indeed, the predictive ability is not good either. And when Amit Khera published this paper, interestingly, even though the title says prediction, they never published anything about the area under the curve or even let alone the explained variance. So we calculated ourselves. So they had a score that was based on data of 2 million variants. It did not include 2,000 variants, but it was based on that. It explained 7%, so definitely better than before. But the reality is that the area under the curve is 0.64. It's like indeed much better than FTO alone, but definitely not good enough for prediction. And these are actually it's like a visualization of the area under the curve. In red is the 0.74 from with the 32 variants. In green, the 0.6, and in blue, just a bit better, the area under the curve for the Amit Khera score. I mentioned you before, we are working with a giant consortium on an even bigger meta-analysis, and if we calculate a PRS on that score, we can explain 14%, so definitely a doubling of the explained variance. But again, here we do improve the area on the curve. It gets closer to 7, so it's not too bad, but it's still not good enough to predict obesity. I can add that instead of trying to predict obesity, if you try to predict extreme obesity, then actually the curve gets a little better. So the more extreme the phenotype, the better the prediction. But all in all, it's still using genetic scores to predict obesity will lead to a lot of misclassification. And maybe one of the reasons why there's so much misclassification is because the phenotype, body mass index is an extremely crude index. Like you and I may have exactly the same body mass index and we may both be obese, but our body composition may be totally different or very different. Our risks for disease, for comorbidities may be very different. The underlying biology may be very different. The trajectory, how we got to the obesity may be very different. I may have been obese as a kid and kept the weight on, whereas you may have slowly accumulated weight over the years. Yet in these GWAS studies, all these factors that cause heterogeneity are totally ignored. If you have the same body mass index as someone else within GWAS studies, these are considered as the same data points. And maybe that's a problem. Maybe that's one of the reasons why it's been so hard to do the translation from variant to function, because the phenotype is so heterogeneous and there's a lot of noise to it. And maybe it's also the reason why the prediction of obesity is so hard, because again, it's that heterogeneous phenotype. And over the years, we've done GWASs. Instead of using body mass index, we looked at body fat percentage. We've used subcutaneous fat as outcome, visceral fat. We've used leptin as a biomarker. So there's different ways to sort of try to reduce the heterogeneity. But one other way of doing that is, for example, by subtyping the population. Yeah. So the idea here is that we have a population with individuals who all have obesity, but the reality is that you can identify subgroups of the population that have similar features. And if you then can identify these subgroups, we call that precision diagnosis, then it will help you with precision prevention, precision prediction, precision prognosis, precision treatment, meaning that, if you know what the features are of this group in Black, for example, then maybe it's also easier to design preventive strategies because you know what the features of this group are, and the same with prediction and so on. And if you can subtype your population, then it might also help with the gene discovery and the translation from variant to function. And of course, in my field, the subtyping would be based on the genetics. So can we identify shared genetic signatures in the population that subset the population? So let me try to explain that a little better. Well, first of all, I should say, like, subtyping is not new in obesity. We've been subtyping based on the phenotype. Yeah? The classical example is people talk about healthy obesity. Like, always be careful with saying healthy obesity because there seem to be a lot of opinions about using that word, but that's at least one way of subtyping, or that's a way of subtyping that people have used. There's other ways of subtyping. People have used machine learning with based on the risk factors of obesity, or I should say more the comorbidities like lipids, diabetes, hypertension, and have identified within a population of obese individuals, some individuals who have a higher risk of diabetes, others have a higher risk of PCOS, others have a higher risk of dyslipidemia, and so on. And then another form is subtyping based on the hedonic causes. This is, it's like, always find it an interesting way of deep phenotyping a population of individuals with obesity based on a variety of hunger hormones, response to certain the gastric emptying, and so on. So Acosta et al., Andres Acosta, identified in his population of obese individuals who are obese because of emotional hunger, because of hungry brain, he summarized, and hungry gut, and slow burn. And I'll come back to that later, actually. But anyway, this is to summarize that this is subtyping based on the phenotypes. What I want to do is subtype a population based on their genotypes. And I'm going to give you two examples. First example is a composite genotype phenotype. What I mean with that is that instead of just focusing on BMI, we focus on BMI and its comorbidities. So what I did, or what we did, we take the GWAS on body mass index and we align SNP by SNP by SNP with a GWAS, in this case, for example, for triglycerides. And we identify individuals, SNPs that are associated with increased body mass index and lower triglycerides, so opposite of what we typically expect based on phenotypic correlations. And we not only do that for body mass index, but also for body fat percentage and waist-to-weight ratio, and not only for triglycerides, but also for HDL, LDL, systolic blood pressure, glucose, and insulin. So we do 18 pairwise comparisons, and that way we have identified 62 independent loci. And these are the 62 loci. Here, every column is one variant, and the color coding corresponds with the effect. Blue means like a negative association, red means with positive association. And the bottom 3 rows are the association with anthropometric traits. The top 7 rows are the association, or 8 rows are the association with comorbidities. So the way to read it, take for example this mTOR, SNP near mTOR, you see it's associated with body mass index and body fat percentage, not so much with anything else, but it's also associated with lower risk for type 2 diabetes, not what you typically expect. Or this variant near the vascular endothelial growth factor A is associated with increased body mass index and body fat percentage, lower waist-to-weight ratio, increased HDL, so that's good, a lower triglycerides, and a lower risk for cardiovascular disease. And in this case, you can imagine that the association with increased adiposity and lower risk of cardiovascular disease may be mediated by a healthier body fat distribution, fat accumulation at the hips. But what you also see here is that you have 62 loci, and the association signatures are different. And that may be because they each of these variants point to different types of biology. So the point I want to make is that instead of just looking at body mass index, and remember, when we use body mass index, the brain is the tissue enriched, is the tissue that is enriched. By combining body mass index with other comorbidities, if we do the same tissue enrichment analyses, then actually we see that the brain does not show up at all. It's adipocytes, adipose tissue, liver, pancreas. So definitely like a different biology, even though the SNPs are also associated with body mass index. Yeah. Just by aligning them now with comorbidities, we get a very different biology that's popping up. Now, we're still looking at subtyping the population, and in a way, this is already subtyping. But we try to do this, take this a little further. And we did a cluster analysis across these 62 loci. We identified 3 clusters. And I admit these clusters are not super strong, and you can already see by eyeballing that cluster 1 may be a good cluster, and cluster 2 a reasonably good cluster. Cluster 3 is a little weaker, and it seems to combine a lot of variants of all kinds of association signatures. But if we then make scores of cluster 1, cluster 2, and cluster 3, then we basically confirm their effect. So the score of cluster 1 associates with, as expected, body fat percentage, body mass index. And what stands out here is their association with body fat distribution. It's like increased gynoid fat percentage, lower waist-to-weight ratio. So cluster 1 seems to be associated with an increased adiposity, but a healthier body fat distribution, and has a pretty big effect on the lipids, triglycerides, HDL, and to some extent also the glycemic outcomes. Cluster 2, as I mentioned, not that strong, has an effect on body fat percentage, body mass index, maybe a little effect on gynoid fat, but not as pronounced as cluster 1, and has some effect on triglycerides and HDL mainly, but not as strong as cluster 1. And then cluster 3, has an overall effect on adiposity, not on body fat distribution, but has a strong effect on the glycemic traits. Yeah? So you see that each cluster has its own signature. So in theory, what you could do is now calculate the score for each individual of your population. Yeah? For cluster 1, you get a normal distribution, for cluster 2, and for cluster 3. And what is of course of interest is the individuals at the extremes of the distribution, because they are most at risk or most protected in this case. So take the individuals in pink are the ones that are in the top 10% of cluster 1, in green in the top 10% of cluster 2, and in blue in the top 10% of cluster 3. So in theory, you could say like, well, these individuals in pink are predisposed to high adiposity, but very low triglycerides and very high HDL. In cluster 2, they are predisposed to high adiposity, low triglycerides, low HDL, should be high HDL, sorry. And in cluster 3, these are predisposed to high adiposity, but low glycemic, low glucose and low HbA1c. Yeah. So the advantage, of course, is of using genetics is that you could already do that at birth, in theory. So you could already predict who would belong to these clusters and what their phenotype might be. And I've been very careful and saying in theory, and the reason why I say it's in theory because the scores that were generated here show very nice association, but to do these kind of predictions, they're not precise enough. Yeah, if we do these subtyping with these clusters, a lot of individuals will be misclassified. But I do believe that if sample sizes increase and maybe if we improve the methods further, then to some extent we may be able at an early, basically at birth, be able to identify some of the subgroups already, which could help with prevention and treatment. I want to give you just one more example. Try to do that quick. We're working on a new GWAS study for body fat percentage and for fat-free mass, like in one go, or I should say 2 meta-analyses, but in one study. We use the data of UK Biobank together with other data that we've previously collected. The GWAS for body fat percentage identifies 685 loci for fat-free mass index, which is sort of a lean mass metric. We identified 773 independent loci. Together, we have about 1,200 loci, but there's an overlap of 209 variants. Overlap in the sense that they reach genome-wide significance for both phenotypes. And in a way, it's not surprising. We know that there's a positive correlation between body fat percentage and fat-free mass. But surprisingly, or maybe not surprisingly, but interestingly, is that not all variants that are associated with increased body fat percentage are associated with increased fat-free mass index. Actually, some show an association with lower fat-free mass index. In fact, that's shown here in this graph. So what you show here is the effect size of the 1,200 loci with body fat percentage on the y-axis, on the x-axis, and fat-free mass index on the y-axis. So in pink, you see the variants that are associated with increased body fat percentage, but not with fat-free mass index. Okay, that's not unusual. In yellow are the variants that are associated with increased fat-free mass index on either side of the body fat percentage, but not with body fat percentage, not significantly. In green are the variants that are associated with increased body fat percentage and increased fat-free mass index, which is almost what we expect. And you see like FTO is one of these, MC4R, TMM18, these are some of the well-known obesity-associated loci. But then in blue, maybe the more interesting ones are the ones that are associated with increased body fat percentage, but lower fat-free mass. Somehow there's the ADAMTS genes. We've not labeled them all, but here's at least 2 of them that were labeled, but other ADAMTS genes show up here, and we don't quite know how to really interpret that. But you can see from this plot that there's some sort of clustering in terms of SNP sets. So what we then did is we did a more formal clustering. So we took the 1,200 variants and we aligned them according to the body fat percentage increasing allele. So that's why they're all associated with they all show like a red square. And we not only did the clustering with body fat percentage and fat-free mass, as shown in the previous plot, but we also aligned it with body mass index, with height, and with waist-hip ratio. And doing so, we identify 6 clusters. The first cluster shows association with body fat percentage, because that's how we aligned it. But at the same time is associated with lower fat-free mass index and lower body mass index, and not so much an effect on height and waist-hip ratio. So we call that cluster the skinny fat. 136 of the 1,200 SNPs go into this cluster. The next cluster is pretty small. It's very similar to cluster 1. The difference is that it's also associated with greater height So we call it skinny fat tall. This third cluster is also small, is associated with increased adiposity, fat mass index, fat-free mass index, body mass index, a little bit with height, but shows an association with protective waist-hip ratio, so optimal body fat distribution. The fourth cluster has large effects, big effects on fat-free mass index, body fat percentage, BMI. A fifth cluster is associated, similar to cluster 4, but not as pronounced, and is associated with shorter height. And the sixth cluster, which is the biggest cluster, is almost more than half of the variance, is associated with increased adiposity, increased BMI, fat-free mass index. So it's sort of the cluster that you would normally, it's like, I think what we normally describe in GWAS studies. So the more interesting clusters, I would say, are this cluster 1 to 5 that we had not identified before. So if we take this cluster now and we make scores and we associate these scores with disease, then you see that cluster 1, and remember cluster 1 is the variant associated with, is associated with, we call it the skinny fat. Then we see that even though it's associated with increased adiposity, it's not associated with any of the diseases. Cluster 2 does not show strong association either, maybe some effect on type 2 diabetes, maybe coronary artery disease, but not very pronounced. Cluster 3, the interesting one, where it's associated with optimal body fat distribution, that despite increased adiposity, is associated with lower risk of these diseases. Cluster 4 has a big effect on type 2 diabetes, probably goes along with a big effect on body mass index, same for coronary heart disease. Cluster 5, similar, but not as pronounced as cluster 4, and then cluster 6 shows association with these diseases as well, but not as pronounced as cluster 4, for example. So then we took these scores and we tested, like, are these scores already associated with adiposity in childhood? So we were fortunate to work with an ALSPAC study who have data from birth up to age 25. And here you see the association for each of the 6 clusters across the life course for body mass index. And cluster 1 is the skinny fat. You see that there's no association of cluster 1 with body mass index in the first 25 years of life. Cluster 2, not really either. Cluster 3 either. Cluster 4, it's like the one that has big effects, already has big effects at an early age. Cluster 5 has an effect, but not as pronounced as cluster 4, and cluster 6 similar actually to cluster 5. So you see that some of the features, particularly for cluster 4, that we see here in adults is already present at a young age. But it gets more interesting when we look, for example, height. There's 2 clusters, cluster 2 and cluster 5, that show association with height in adulthood. That is already visible clearly here at a young age. Like basically immediately after birth, we see that genetic predisposition to greater height is visible for cluster 2, and at the same time for cluster 5 as well. They have some information on gynoid fat accumulation, and one cluster that we're particularly interested in seeing an effect is cluster 3, of course. It has a big effect in adulthood on waist-hip ratio, and indeed, of all the clusters, we see that at a young age, it has an effect already on gynoid fat accumulation. Just to bring things back, because in the end, we want to use genetics to subtype individuals with obesity. What we currently do is we have a group of individuals, and when we do these weight loss interventions, whether it's behavioral weight loss interventions, medications, we actually don't really make a distinction. It's like we prescribe the same drug, and some individuals lose weight, some individuals don't really lose much weight, and some individuals may even gain weight. But what we really want is that before we actually prescribe medication, we identify these subgroups. I give you an example of screening them for genetics, and based on the genetics, we create the subgroups. But as I said, genetics is definitely not the be-all and end-all. You need other factors to better subtype them. But in the end, what you want to do is identify these subtypes. Is it with genetics or other omics? to then prescribe a more tailored weight loss intervention. I like genetics in particular, and I would love for it to work, because the big advantage of genetics is that it's already there at birth. The big challenge with other features, particularly in the context of obesity, is that omics, many omics, proteomics, metabolomics, microbiomics, they're not the cause, or it's very hard to determine whether there's any proteins or biomarkers that are causal to obesity. It's often that obesity has changed the proteome or the metabolome. So it's very hard to sort of tease out what's causing consequence. I want to just bring it back to just give you an example of what might be the dream. So I mentioned to you that Andres Acosta and colleagues had identified these 4 subtypes in a previous study in 2015. And in a very recent study, what they did is they used these 4 phenotypes to determine if you belong to one phenotype, they would then prescribe a specific weight loss medication that's different than if you belong to another phenotype. So they tailor the weight loss medication to the phenotype you belong to. And then they indeed show that if your medication was prescribed based on your subtype, then the weight loss was much more pronounced than when you get the one-size-fits-all prescription. That's what you see here in red. Yeah. So this is the hope and the expectation that one day, even maybe with genetics, we can get results like this, at least in the context of weight loss here. So just to summarize, we identified, and this is a typo, almost more than 1,100 genetic variants. And as you see that with the giant consortium, we're expecting that many more variants will be identified. The subtyping of obesity is at its early stages. I gave you an example where we try to use the alignment of GWAS for body mass index with comorbidities to identify these subtypes, with the hope that it could contribute not only to the precision medicine in general, but also to the gene discovery and translation. Big advantage of genetics is that it's accessible early on in life. It may represent biology, may facilitate translation, and so on. But just to be very realistic, I also know that obesity is a complex disease and genetics is not the be-all and end-all. So for anything we do in the context of precision health, genetics is only the starting point or one of the contributing factors, and we will always have to include non-genetic factors. I think I will finish here, and hopefully there's still time for some questions. I'm more than happy to stay after. I'd be very grateful to my group here in New York, and also my growing group in Copenhagen, to the many collaborators, in particular the Giant Consortium and funders here in Copenhagen and the NIH, of course. And I'm happy to take questions.
Thank you very much, Ruth. This was really fantastic. I really enjoyed especially your genetic methods for self-certification of the phenotype. The session is open to questions. Please feel free to type your questions in the chat, or you can simply raise your hand and unmute yourself. So perhaps as we are waiting, I'll just start with a quick question. So, you know, ancestral diversity is a kind of an issue for a lot of early GWAS studies, which were really dominated by European ancestry, and that also affects kind of the transferability of polygenic scores. That probably also applies to polygenic scores for obesity. Do you know the kind of ancestral breakdown of the latest Giant Consortium GWAS, and to what degree, you know, the global diversity is actually represented in the latest study?
Yeah, you know, we do better than we've ever done before. We have a good, and I don't know the exact percentages, I should know, but we have a good representation of the Asian population. We have, thanks to 23andMe, we have a good presentation, and MVP, we have a good representation of the Hispanic population. Still low representation of African American population still, but it's better than we've been able to do before. Yeah. but it remains an issue, or an issue, it remains a challenge to do the transferability across different ancestries.
Great, thank you. And we have a question. Dan, if you want to unmute yourself and ask the question in live, feel free to do so.
Yeah, I was just curious, you showed a lot of clusters, and I was wondering if there was any specific genes or pathways, mechanisms that you saw when you were looking at those clusters.
Yeah, you know, when we do these 62, the healthy obesity, let's call it the healthy obesity, 62 healthy obesity loci, we definitely see different genes, or like when we do prioritization, different genes than when we look at just body mass index in general. The clusters, comparing the clusters is hard because they were not solid enough to really say like this is a different biology. What we did do is the same tissue enrichment analyses that we did in the 62 loci. We did it cluster by cluster. What we found is that the adipose tissue, adipocytes, liver, pancreas, that the enrichment, if we just restrict it to the loci in cluster 1, it's even more significant than when we do it across the 62. So it's seen that this is a more homogeneous cluster. But in cluster 2 and in cluster 3, nothing in particular showed up. We did not have enough power. So we would suggest that cluster 1 is maybe the better, stronger cluster to follow up. I know you're asked for specific genes. It didn't really, yeah. Yeah, not specifically for the by cluster. Yeah. We did see some genes involved in the beiging of the brown adipose tissue across the 62 loci. It's like very different from when we look at BMI associated loci as such. But otherwise, I would not be able to say which specific biology shows up.
All right, there's another question from Eva. Eva, feel free to unmute yourself, or I'm happy to read your question, but perhaps you can just ask it. Maybe I'll just read it. Yeah. So what do you think about the challenge of gene by environment interactions in the GWAS? Do we have enough power to predict the interactions and to perhaps include them in precision nutrition?
Right, that's a good question. You know, we've done these GWAS studies where we, at the genome-wide level, look for variants that of which the association with body mass index is different based on like physical activity status, smoking. I think indeed some dietary factors have been tested as well. And actually we don't, even though we have huge sample sizes, hundreds of thousands of individuals with that data, we don't find evidence of interaction. It does not mean that there is no interaction. It's just that maybe GWAS is not really the method to test for these interactions. I think the only locus that seems to come up in these interaction studies is FTO, so that the effect of FTO is more pronounced when you live an unhealthy life. Taking that to precision nutrition and it's like what 23andMe does, gives recommendations based on your genotype. It's actually, if we cannot identify it with GWAS studies and 100,000 of individuals, then it probably also means that you cannot use the genotypes to prescribe which diet is good for you. Whatever these online companies do, it's probably like very imprecise.
Okay, I see another question from Rudy. Rudy, please go ahead and ask your question.
Yeah, Ruth, thank you very much for these heroic studies. I'm curious about your insight into the following sort of inference from some of the clustering that you showed with regard to control of body shape and its influence on some of the comorbidities. And clearly that's true. And I'm very curious about, the fact that some of that shape is incurred during puberty. It's not so apparent in a pre-pubertal individual but becomes obviously much more apparent afterwards. What can you say, if anything, about the genes that might underlie that particular interaction of puberty with body shape? Because those genes might be the ones that are most dispositive regard to the creation of body shape and thereby mitigate or mediate some of the effects on degree of adiposity by shape. I'm really curious about the genes that actually, as you pointed out early, the BMI looks like in the aggregate, it's driven by brain, but the susceptibility to some of the consequences is not driven by those genes, but some of the others that confer this shape phenotype, blah, blah. So it's a tough question. But.
Yeah. So the thing is, we did not, the Alspec study did not have data before puberty. So it's hard to say that it only shows up at around puberty or after. It could be that's actually already present before.
Well, that's why I asked you, because it looks like you have some data prior to puberty. And so you might expect to see some interaction, or at least some predictive power there.
No, for body shape, we don't have that early data.
Okay.
Yeah, but the genes, it's the same as these healthy obesity genes, like mTOR, vascular endothelial growth factor, PPAR gamma, sort of the genes that act at the adipocyte level, I would say adipocyte biology level. Definitely not, the brain is not involved there. So it's more peripheral biology.
Yeah, I agree. I'd like to know what those are. That's all. I mean, whether you can identify ultimately which are the predisposing genetic variants for those phenotypes. That would be big important to know.
Yeah.
So there's another question, and just in the interest of time, I will just ask the question. So do you think there's a role for genetics in predicting who would best benefit from bariatric surgery?
So they've used PRSs for that. For example, to say like, if you have a high genetic risk score, maybe like you'll be resistant to this surgery or any other intervention really. And that's not clear. The evidence that if you have a high risk score, you would not respond to bariatric surgery, it's not confirmed. And even if there was an association, say that indeed the people with high genetic score show less weight loss, I don't think you can use it really to predict who should get bariatric surgery and who not. Again, because obesity is more complex and it's more than just genetics. So we need other factors to supplement the genetic information. So I don't want
maybe I can just squeeze in one other question, and that is kind of asking for your expert opinion on the identification of or prioritization of causal genes within GWAS loci. Obviously, that's a very active area with a lot of new methods in development. Do you have a favorite method, or is there? Do you have a sense if there is any new transformative technology that can help with this issue?
I always say nearest gene.
Yeah, that's pretty much what people have been using right all along. Clearly, it seems problematic with FTO, but yeah, that seems to be consistent in the predictions. But do you feel there's anything beyond nearest gene?
I think you need to use multiple methods. Like each method has their own thing and like a specific feature where it focuses on, and it works for one locus but not for the other. So I really think you need a combination of methods.
Great, thank you so much. We are out of time, but this was fantastic, Ruth. Thank you for accepting our invitation. And I know it's really late at night in Copenhagen, so good night. Thank you.
Take care. Bye-bye.